Good morning, everybody. This is the last Shanks Foundation sponsored PathSoc Grand Rounds lecture of 2025. For those who don't know about these lectures, they occur most months and are kindly sponsored by the Shanks Foundation. And they're also available online on the PathSoc website and also on YouTube. It gives me great pleasure this morning to introduce Adam Byron, who is a lecturer and group leader in the School of Biological Sciences at the University of Manchester and is a member of the Manchester Cell Matrix Centre. So Adam's work concentrates on the tissue microenvironment and how it elicits signals at the nucleus of cells and the control aspects of cell behavior, including regulation of gene expression in both health and disease. Adam studied biochemistry at the University of Warwick and then spent some time with AstraZeneca before he did a PhD in biochemistry. Some of his mentors include Professor Martin Humphreys and Professor Margaret Frame, who's based in Edinburgh. So since 2022, Adam returned to Manchester and is an independent researcher and will be speaking today about his work linking the extracellular matrix to gene regulation. So without further ado, I will hand the lectern over to Adam, and thank you very much. We're really looking forward to your lecture. Thank you.
Thank you, Sarah. Can I check everyone can hear me okay?
Yeah, sounding good.
Thank you very much. Thank you for that kind introduction. And thank you also. It's a great privilege to be able to contribute towards the Pathology Grand Rounds. So as Sarah kindly introduced, I'm a group leader at the University of Manchester, and we're very interested in how cells interact with their surroundings and how this contributes to control of cell behavior and dysregulation of cell behavior in cancer. And in particular, in this talk, I'm going to be focusing on how changes at the cell surface could potentially influence the transmission of forces and biochemical signals to the nucleus to control gene regulation in cancer. So in our group, we're very interested in how cells interact with their surroundings. And often these interactions are happening at the cell surface, the interface between the cell at the membrane and the extracellular matrix. And for many years, we were interested in the molecular components that regulate this interface. So we spent some time developing approaches, especially proteomic approaches, to characterize the molecular complexes, the molecular machinery, essentially, that is involved in sensing and responding to changes in the surroundings of cells in the tissue microenvironment. Just going to pop a pointer on, hopefully. Here. So more recently, we've also been starting to realize that many of these proteins that are typically found at the cell surface involved in cell adhesion processes can also be found in other parts of the cell. And so we were particularly interested to note that a subset of adhesion proteins can translocate into the nucleus in certain settings of cellular stress, including in cancer. And there they can regulate a range of processes, including chromatin organization and the transcription of genes. And so today I'm going to talk to you about the new roles for some of these adhesion proteins that we've started to identify in the nucleus, and how these may be really important for controlling the expression of cancer genes, especially linked to the matrix and the immune system. So I'll begin the story back in work that was ongoing in Edinburgh some years ago, when we were interested in this adhesion kinase called focal adhesion kinase, or FAK. FAK is strongly upregulated in many solid tumors, and work by Alan Serrels and Tom Lund in Margaret Frame's lab was investigating the role of FAK in tumor growth. In this example here, we've got a cutaneous squamous cell carcinoma mouse model in which the cells that are normally expressing FAK can be grown as subcutaneous injections in the mouse, and they form tumors. But Tom and Al noticed that in the absence of FAK, or even in the absence of a nuclear localization signal that they had identified within the sequence of FAC, removal of that alone was sufficient to cause regression and clearance of the tumors in this mouse model. And they also noticed that this had a key link to whether the immune system was functioning or not. So these experiments on the left are the same as these experiments on the right, done in the FVB syngeneic host model. But when they repeated the experiments in a mouse model that did not contain a fully functioning immune system, the ability of the FAC null tumors to be cleared was lost. So this gave a clear indication that a functioning immune system was really important for the role of clearance of these tumor cells. So I wanted to understand why that might be. So we did some gene expression analysis of the cancer cells that either contained FAC or did not contain FAC to try and identify what those differences might be, because we knew that FAC was in the nucleus. So our hypothesis was that it might be regulating gene expression there. And we found that in the cancer cells, FAC appeared to be driving a subset of chemokines expressed from the cancer cells, for which the cognate receptors of those chemokines were also upregulated in immunosuppressive Tregs that had infiltrated the tumor microenvironment, suggesting to us that there was this key chemokine ligand receptor axis that was upregulated that was creating a tumor, an immunosuppressive microenvironment in the presence of FAC. Whereas in the absence of FAC, this axis was lost. And Tom and Alan showed that there was a concomitant infiltration of CD8 positive T cells into the tumor microenvironment, which was responsible for this clearance of the tumor cells. So this really highlighted to us that a nuclear adhesion protein, in this case FAC, was really critical for controlling gene expression that could have a knock-on impact on the tumor immune microenvironment. And work by Marta Canal and others has gone on to show that FAC has other roles in regulating the immune response. In this case, in pancreatic ductal adenocarcinoma, it plays a key role in regulating proteasomal processing of antigens and also presentation of antigens to the immune system, suggesting that FAC has other additional roles in regulating the tumor anti-tumor immune response. But if we look at the literature, it's not just FAC that has been noted as an adhesion protein to have roles in the nucleus or with nuclear functions. So back in the 90s, Mary Beckley was describing Zixin as a really important potential shuttler between the plasma membrane, the sites of adhesion, and the nucleus. And subsequent work has illustrated similar phenomenons for other adhesion proteins, some examples listed here. So we were really keen to understand to what scale adhesion proteins may have roles in the nucleus. So I began by looking at the plasma membrane side of the coin. So this is looking at isolating adhesion complexes, or integrin adhesion complexes, abbreviated to IACs, from cancer cells and other cells to try and characterize the composition of the adhesions by mass spectrometry. So we've developed several different approaches to do this using proteomics. And when we compiled multiple data sets, this is work done by Ed Horton and myself in Martin Humphrey's group, we can map the known localization of the entire list of components from the adhesion complex. And many of the zones to which these proteins localized were expected. It was to the cytoskeleton, to the cell junction, to the edge of the cell, and also involved in vesicle trafficking, so the recycling of these components back to the cell surface. But we consistently identified a subnetwork of proteins that had also been previously described to localize to the nucleus, suggesting that there was certainly a component of this adhesome that could have roles in the nucleus. And this is another way of looking at this from a more functional point of view. When we map functions of all the proteins, we can say this wing of expected cell adhesion proteins, functions linked to vesicle trafficking, but also this part of the network that has proteins with annotations linked to nuclear processes such as DNA organization. It was interesting to note that many of these proteins contained limb domains. So limb domains are known to be involved in nucleocytoplasmic shuttling. Many of the proteins that Mary Beckley were describing in a couple of slides ago do contain limb domains, such as zixin, which is the example I mentioned earlier, but also paxillin and others. Many of these limb domain proteins also have been described to have nuclear export sequences. Some of them have nuclear localization sequences. Some of them have both. Some of them are less well described. So there was a theme emerging that the adhesome is enriched for limb domains, which can bind cytoskeleton, but also the property of these limb domain proteins is the ability to shuttle to the nucleus. So we then flipped the coin and wanted, when I was in Margaret's group, wanted to understand, well, in the nucleus of cancer cells, which adhesion proteins can we detect there to potentially catalog adhesion proteins that have the ability or propensity to localize to the nucleus. So to do this, we again leaned on proteomics. So we developed an approach to really purify nuclei away from the rest of the cell, including cytoskeleton and perinuclear organelles that normally come down with nuclear preps. To try and purify nuclei from, again, we're working with this SCC, non-melanoma squamous cell carcinoma skin cancer model in which we know FAC is nuclear. And we use proteomics to characterize the adhesion proteins, which we'd already developed lists of from our work with Martin, to characterize adhesion proteins that can localize to the nucleus of the SCC cells. And there were several themes that emerged. We could cluster the proteins by the canonical adhesome axes. This alpha-actinin module is enriched for actin-binding proteins, but also you'll note by the small gray circles that several of these proteins also contain limb domains, as we'd observed from the larger scale data analysis previously. Another emergent theme was FAC-associated proteins, and several of these contain firm domains. So a firm domain, FAC contains them, these other proteins here also contain them. They're important for proteins to bind to receptors such as integrins. But also, intriguingly, firm domains often have a nuclear export sequence within them. And we found that a large proportion, over half of the firm domains that are known to be in adhesions, were also in our nuclear fractions in these cells. So we could use network and graph-based approaches to expand this network to known adhesion proteins, which also then brought in other adhesion proteins that had not been previously described to be in the nucleus. One example is this tumor suppressor testin, and we could go in and see in cancer cells there was some, so the black staining here is the co-localization of the nucleus in green and testin in magenta. There was some localization in these cancer cells, but when we stress the cancer cells, in this case with an oxidative stress using hydrogen peroxide, there's a striking enrichment of testin in this case into the nucleus, reinforcing this idea that under stress conditions, adhesion proteins appear to be entering the nucleus to control functions. So talking about functions, we wanted to really start to then dig into moving beyond what we characterizing the list of proteins that are in there to what they may be doing in the nucleus. So for this, we started out by using a combination of transcriptomics and proximity labeling interactomics. So we had several adhesion proteins tagged with proximity labeling enzymes that essentially label with a biotin and moiety any other proteins that are in the vicinity. So we can capture that so-called proximal interactome. And we did this in the nucleus for several adhesion proteins. And intriguingly, we found that there was a large overlap in the adhesion proteins that were interacting in the nucleus. So this really suggested to us that these adhesion proteins, as they do at the plasma membrane, may also be working together in complexes in the nucleus. And intriguingly, we found that different adhesion proteins that we could see in the same proximity in the nucleus were regulating the same sets of genes. So if we stopped FAC going to the nucleus, or if we deleted FAC, there was a downregulation of these two exemplar genes and an upregulation of PDGF receptor beta. And we saw exactly the same patterns if we knocked down HIC5, which is also a member of this complex in the nucleus. So this suggested to us that adhesion proteins were working together in the nucleus to co-regulate genes in these cancer cells. So the next part of the story moves to another adhesion protein that we find having an important role at the nucleus. And this is a cytoskeletal regulator called MENA. So this is a member of the enabled ENA-VASP family of proteins. It was described by Frank Gertler in the 90s and has since been revealed to be very important in breast cancer progression and other cancer progression as well. And in particular, Frank Gertler's group and Paolo Nistico's group have been key in revealing seminal discoveries about the splicing of MENA and how different spliced isoforms of MENA can drive different properties, from an invasive mesenchymal like phenotype with certain invasive isoforms to more epithelial characteristics in an isoform called MENA 11A. And how do we get to MENA? So again, we were interested in the composition of adhesion complexes and how that may overlap with potential nuclear functions. And so for these experiments, we switched to patient-derived cutaneous squamous cell carcinoma cells in collaboration with Irene Lee at QMUL, who was at Dundee at the time. And we're now carrying on this work with Gernot Walko in London. And they have a fantastic cell and tissue bank of patient samples from various squamous cancers, but particularly cutaneous squamous cell carcinoma. And this is great because we can access samples from patients that present with a primary tumor. There are often then these tumors often recur after surgery, and then we can get samples from those recurrences and ultimately from, in this case, from lymph node metastases. So we're able to isolate isogenic sets of cell lines from the same patient during the cancer journey. And so the data I'm going to show here compare SCC cells from the same patient, comparing the primary tumor with the lymph node metastasis. And so again, we moved to our proteomic, our favorite proteomic technique to isolate adhesion complexes from the cell matrix interface of these patient-derived cells, and using proteomics to interrogate the networks that form there. And so this was work in Margaret's lab with Fred Lee-Mo Chi, who drove a lot of this and built in interaction network modeling into the analysis of the proteomic data to really try and extract functionally relevant communities of proteins from these proteomic data. And so what these analyses reveal, firstly on the left, are subsets of functionally related proteins that are identified in the adhesion complexes of these cells. You can see a highlighted example here is an actin regulatory module, which I'll come back to shortly. On the right are the same data, but as a network view. And here we've mapped on the changes in abundance of these proteins, comparing the metastatic cells, which are in red, versus the primary tumor cells, which are in blue. And so you'll note that this actin regulatory subnetwork is strongly enriched in the metastatic cells. So these are proteins that are being recruited to the adhesion complexes more in a metastatic cell. And so if we zoom in on this subnetwork, we were intrigued to find that it was dominated by this actin regulator MENA, really strongly enriched in the metastatic adhesion complexes. There were other kind of actin regulators in this module, but we were really intrigued to find this actin binding, but normally nuclear membrane associated protein called Nesperin-2 as part of this subnetwork. So we wanted to ask the question of how does MENA link to a potentially nuclear membrane protein? So we started by looking at where MENA localized in these patient-derived cells. MENA is in magenta, so you can see on the left here orange arrowheads pointing to where one would normally observe MENA, strongly enriched in focal adhesion structures or adhesion complexes that are at the plasma membrane interfacing with the extracellular matrix. But when we co-stain with Nesperin in green, we also find a striking localization of MENA around the nuclear envelope, which is marked here by Nesperin. You can see quantification of that co-localization of Mina and Nesprin at the nucleus. So it wasn't that Nesprin was necessarily enriched in the adhesion complexes here. So Fred went on to do more high resolution imaging. In this case, this is structured illumination microscopy sim imaging. And this is now a slice down through the cell. So this is the basal side here. This is the dorsal side of the cell here at the top. And again, we've got Mina in magenta, we've got Nesprin in green, and also actin in blue. And when we zoomed in on punctae of Nesprin, we found that just dorsal to those Nesprin puncta, likely marking the nuclear envelope, where there was Mina staining co-aligned with those Nesprin puncta, and also that also co-aligned with actin staining as well. And we know that Mina and Nesprin both bind actin. And so intriguingly, this just to go back to the patient data as well, there was a much stronger co-localization of Mina and Nesprin in the metastatic cells as compared to the primary cells derived from the primary tumor. So we started to think about how we can put this into a model to build our hypothesis. So we know Mina is an actin binding protein. We know that Nesprin also binds actin at the nuclear envelope. So here in this box, we've got a zoom in of the nuclear envelope. These are nuclear pores. This is the nuclear membrane. And Nesprin is a key component of this transnuclear envelope spanning complex called the link complex. Now, the link complex is very important for connecting cytoskeleton through the nuclear membrane to the nuclear skeleton composed of lamins. So we wanted to understand, well, we've seen by imaging that Mina can co-localize at the nuclear envelope. It appears to be in proximity to Nesprin there. Does it indeed interact with Nesprin? And so we were grateful to Maddie Parsons for some Nesprin fragments, GFP Nesprin fragments, to start to interrogate this interaction. Nesprin's a very large molecule, and we wanted to understand, does Mina bind and where on Nesprin may it bind? So using a range of different Nesprin fragments, we were able to pin down a potential interaction of Mina to this region containing spectrin repeats 49 to 56. So you can see here the span of Nesprin, and 49 to 56 is in this C-term, towards the C-terminal of the molecule, which is where Nesprin is interacting with the nuclear envelope via its cache domain. So we think that Mina is interacting with Nesprin quite close to the nuclear envelope. So we wanted to know how does that potential interaction between Mina and Nesprin control the function of the link complex, or at least in the first instance, the ability of the link complex to bind to what it would normally bind. And so, as I mentioned, Nesprin through the link complex binds to actin. So we knocked out Mina using CRISPR in the patient-derived cells. And we find that where normally Nesprin is able to interact with actin, in the absence of Mina, that ability to interact with actin is dramatically reduced. And when we reintroduce Mina exogenously into these cells, we actually slightly overexpress Mina into these cells, or considerably overexpress Mina into these cells. And that results in an upregulation then and a rescued, an over-rescued almost ability of Nesprin to interact with actin, suggesting that Mina is key for this link between actin and Nesprin. Nesprin is also important via the link complex for interacting indirectly with lamins in the nuclear skeleton. So we wanted to test, does that potential interaction with Mina influence how the link complex can connect through to the nuclear skeleton? And so we used the same approach. We used an IP of Nesprin and looked to see whether it could interact with lamins. So lamin AC usually interacts with the link complex using this approach. And we found that there was a reduction of lamin AC interaction with Nesprin that could be rescued when we reintroduced Mina into the system. So this suggested to us then to expand that model, that in the absence of Mina, there is a decoupling of the connection between the link complex and the cytoskeleton. And also that connection results in a poorer interaction of the link complex with the nuclear skeleton. So this has important functional implications for the cell. Firstly, the cell is, the link complex is connected through to the cytoskeleton, which transmits force essentially all the way from the extracellular matrix through integrins that are connecting through to the cytoskeleton, transmitting the tension that can change when extracellular matrix properties stiffen or they change their viscoelastic properties, which you often see in fibrosis and in many cancer solid tumors. So that force is being transmitted all the way through the cytoskeleton to the nucleus, and the link complex then is really important for transducing that force into the nucleus. And the link complex associated protein emrin has been described as very important for converting that mechanical force into a biochemical signal. And under tension, the link complex senses this tension, and that is read out by the tyrosine phosphorylation of emrin. And in the absence of force, there is a decrease in the phosphorylation of emrin. There's a decrease in force transmission across the link complex. So we wanted to test if MENA had a role in this force transmission and conversion to a biochemical signal at emrin. So to do this, we IP'd tyrosine, phosphotyrosines from the lysate and blotted for emrin. So we were looking for phosphorylated emrin in the presence of MENA, in our CRISPR knockouts of MENA, and then upon re-expression of MENA. And as you can see from the Western blot or from the quantification below, in the absence of MENA, phosphorylation of emrin is dramatically reduced, suggesting that force transmission through the link complex is being perturbed in the absence of MENA. So MENA needs to be there to reinforce that link complex. So it is more of a reinforcement than a requirement, because we still see the link complex connecting through to the nuclear skeleton to a certain extent in the absence of MENA. But I think that MENA is there to reinforce that connection from the link complex to the cytoskeleton to enable force that is being sensed at the plasma membrane to be transmitted into the nucleus to regulate this mechanosensor emrin inside the nucleus. So the next question is, what is the importance of this phosphorylation of emrin in the nucleus upon sensing force? So it's known that in the absence of tyrosine phosphorylation of emrin, the chromatin-associated protein BAF can be recruited to emrin, and it brings chromatin with it. So this heterochromatin recruitment to the nuclear periphery, driven by BAF and accessory factors in the absence of tyrosine phosphorylation of emrin, results in the transcriptional silencing of genes that are within this region that's recruited to the periphery. These are sometimes called laminar-associated domains, or LADs. Whereas when force can be transmitted through the adhesion complex, through the link complex, resulting in phosphorylation of emrin, this reduces the recruitment of heterochromatin to the nuclear periphery and enables these promoter enhancer regions to drive transcription of those genes. So is this the case here? So we began by using our cells that express MENA and have been knocked out for MENA to understand what are the gene expression changes in the absence of MENA. And there were several. Here we've interrogated cell migration genes and genes linked to extracellular matrix organization and the immune response. And in the absence of MENA, all these genes were downregulated here. We've got one example that is the most dysregulated gene in this data set, which was the innate immune component pentraxin-3, which is encoded by PTX3. So we wanted to try and link these changes in gene expression to the mechanism I showed on the previous slide related to reorganization of chromatin. So to do that, Fred Limochi collaborated with Wendy Bickmore's group to use an approach called DAM-ID. So this is, again, a proximity-based approach, but this time we're thinking about chromatin interactions rather than protein interactions. So this technique uses a DNA methylase, adenine methylase from E. coli tagged onto a protein of interest. And in this case, we used lamin B1, which should be at the nuclear periphery. So then in this system, any chromatin that approaches this tagged lamin B1, those adenines on that DNA will be methylated. We can then extract and enrich for this methylated DNA, amplified methylated DNA, and use sequencing to understand which regions of chromatin are close to the nuclear periphery and which are away. And how does that change when we delete MENA from the cells? And so again, focusing on our highly dysregulated innate immune component PTX3, These are tracks from the region around the PTX3 gene. And you can see in cells that express MENA, there is very little association of the PTX3 gene region with the nuclear periphery. Whereas in the absence of MENA, there is a significant recruitment of the promoter enhancer region of PTX3 to the nuclear periphery. And as you'll recall, that recruitment of heterochromatin regions to the nuclear periphery is associated with transcriptional silencing. And as you can see by this confirmatory PCR, that is indeed what we see with PTX3 in the absence of MENA. And indeed, we've gone back to many of those other those other genes that you saw in that volcano plot on the previous slide, and found similar patterns of recruitment. So genes that are downregulated have an increased recruitment to the nuclear periphery in the absence of MENA. This is just some information about PTX3. It's very interesting. We're keen to explore this further. It's got roles in extracellular matrix organization as well as in the innate immune system. It's been implicated in cancer cell invasion, and actually is a pancreatic ductal adenocarcinoma biomarker as well. So just bringing in broader context then, in this case, linking to work from Paolo Nistico's group, investigating MENA in the tumor microenvironment of lung cancer. We know from her work and work that we're starting as well that MENA isoforms are really important here. So we haven't delved into that in the mechanisms that I've just described to you, but we're starting to do so. And Paolo's shown that different isoforms of MENA can have a real impact on the gene expression and the protein secretion of extracellular matrix and immune components. And so in these cases that are shown here, where MENA 11A, which I mentioned earlier as the epithelial-associated, less invasive isoform, where MENA 11A is expressed, there is a downregulation of extracellular matrix and indeed immune genes into the microenvironment. Whereas when more invasive MENA isoforms are expressed, there's an upregulation of many genes, including fibronectin, which we also see in our data set. And not only that, Paolo's shown that the MENA isoforms can also regulate receptors at the cell surface, the integrin adhesion receptors that are binding to and responding to the extracellular matrix are also differentially expressed by different MENA isoforms. And this also impacts whether cells are able to invade to the same extent in an integrin-dependent manner. So we think there's a really important link from MENA to the extracellular matrix and the tumor microenvironment through its control of gene expression. And we're continuing to investigate that now. But we're also very keen to link what we're observing to the extracellular matrix itself and understand how the structure, composition of the extracellular matrix influences cancer cell survival and tumor growth. And so we've developed several proteomic approaches to characterize the extracellular matrix composition of cell lines, but also of tissue. And in this example, this is a non-small cell lung cancer example from patient samples who are undergoing curative intent surgery in collaboration with Asan Akram at Edinburgh. And so work by Helen Titmar showed that if we isolate samples and compare tumor tissue to non-tumor tissue from the same patient, we can identify subnetworks of extracellular matrix molecules and other molecules that are important for regulating the extracellular matrix. In this case, a subnetwork of functionally related proteins that control extracellular matrix cross-linking. These are all red, so they're upregulated in the tumor microenvironment, suggesting that there could be concomitant changes in the stiffness driven by changes in cross-linking of the matrix environment in those tumors. These data sets are also really informative for extracting broader signatures of disease. And going beyond just non-small cell lung cancer signatures, we've actually found that many of these extracellular matrix molecules discriminate between subtypes of non-small cell lung cancer. So we have some signatures that are really informative for adenocarcinoma, and we have others that are more informative for squamous cell subtypes. And we had both adeno and squamous patients within this cohort to allow us to discriminate here. So as I bring the talk to a close, I'll summarize our previous and current and ongoing interests in the lab. And this is really to understand how the cells interact with their surroundings, how adhesion receptors, especially integrins, sense and respond to these changes in the microenvironment, but also how some of these adhesion proteins involved in adhesion signaling are also triggered to relocate to the nucleus, either into the nucleus, as is the case for the FAC example I described, or to the nuclear envelope, as in the case of the cytoskeletal regulator MENA. But ultimately, these proteins appear to be regulating not only gene expression, but also the organization of chromatin itself to influence transcription. And for both the examples I talked about, the genes that are influenced are often secreted gene products. So these are chemokines or extracellular matrix molecules that are then remodeling the tumor microenvironment to control tumor growth or to control anti-tumor immunity. And these actually really will circle back then to the receptors on the cell surface that are sensing these changes. And we're really keen to understand the full circle of these mechanisms here and how they all feed back to each other. So I'd like to acknowledge a lot of people who have been involved throughout this work from the very beginning in Manchester with Martin and especially John Humphreys and Ed Horton, driving the characterization and understanding of adhesion signaling complexes at scale. Then in Edinburgh, I was really fortunate to work with very talented people on skin cancer models and understanding how proteins in these skin cancer models are important in the nucleus to control transcription. And then now I'm now set up in Manchester, pursuing several aspects of this work further to understand the mechanisms involved in regulating gene expression in cancer driven by microenvironmental changes. My lab here are highlighted in blue, past and present, some fantastic work that's been ongoing from Carl Connolly, Izzy and Sylvia, and Josie and Julio have just started embarking on this work in skin and lung cancer. So I'd like to thank the funders for funding this work to date. Really pleased to have just been awarded a BBSRC New Investigator Award, which will kick off in the new year to further grow this research. But I'd really like to thank you for your attention. And of course, I'm very happy to take some questions. I'd be very happy to connect with you during the rest of the call or offline later on. Feel free to get in touch by whichever means you would like to. But thank you for your attention. I'm very happy to take any questions.
Thank you very much, Adam. That was brilliant, a fantastic overview. Very clear and didactic. It was terrific. Just to let everybody know who's online and has been listening to Adam's lecture, there is a Q&A section at the bottom of the screen. So please feel free to ask any questions. So there are a couple already. Adam, if you don't mind, there's one, an anonymous attendee who says, do different cell lines for the same cell type behave differently when it comes to adhesion and the signatures?
So different cell lines from the same cell type. So we have partially addressed that with some of our proteomic work, looking at adhesion complexes from different cell types. And what we tend to find is there are core sets of adhesion proteins that are generally enriched in different cell lines, even actually across different cell types as well. And we've been able to define a kind of a core adhesome that is almost always detected. And then around that core adhesome, we have different modules that are differentially enriched under different circumstances. So these could be in different cell lines from the same cell type. They could be in under different culture conditions. So we think that the complex has this core set of components that need to be there, but the peripheral modules are quite dynamic. They're differentially recruited. And so we think that gives cells different opportunities to respond in different ways as is appropriate for that cell type. So the proteomics has been quite revealing in that regard in terms of compositional dynamics in different cell types.
Thank you. I'll just jump to number 3. They wondered whether you could please share your screen again with the code. the QR code, and so we'll leave it on just a touch longer. And and so so hopefully everybody can see that.
Hang on, that should reveal itself. There we go.
Okay, thank you very much. So Mark Aarons, who I think you probably know from Edinburgh, says, fascinating research, Adam, thank you. What are the opportunities from this work for drug development to intervene in these processes for anti-cancer treatment?
Yeah, I mean, it would be fascinating to think about how to target nuclear localization for sure, given advances in things like PROTACs these days. Certainly, Alan with Stefan in Edinburgh was driving combination trials with a FAC inhibitor alongside PD-L1, so combination immunotherapies in solid tumors. So that was targeting the kinase activity of FAC, but still had an impact on its role in driving transcription of immune components. So that was underway. It would be very interesting to try and redistribute nuclear localized adhesion proteins. Not clear exactly how successful that would be yet, but I think it would be fascinating to try and explore that for sure.
Right. Thank you very much. So there are no further questions, but I still would encourage people who are online to submit some questions. I've got some questions, if you don't mind. Just wondering, have you had the chance to look at the differences between the primary tumor sites and say perhaps the metastatic site or metastatic organ, and are there differences in your integrins and adhesion molecules that may be present, and then also would have significance for any targeted drug therapy?
Yeah, that's a very good question. So for the project I described, we were using the cell lines derived from those sites for the work, but we haven't got data on how that looks in the tissue itself. So that would be really interesting to know, how is that recapitulated. We've got those different sets of adhesion components that are enriched differentially between primary and metastatic. So we've got those candidates that we could, as a starting point for exploring. One thing that Fred noticed in these adhesion complexes was there was a shift in the integrins that were at the cell surface and engaging with the matrix, comparing the primary to the metastatic site. So that suggested to us that the cells they had a different propensity to sense the microenvironment. They had different integrin heterodimers expressed in those two different settings, which would mean that they were then able to bind to different extracellular matrix molecules and signal from those matrix molecules differently, which could be part of the mechanism that's driving those, different responses and different growths that we observe. But we haven't gone back and actually validated that in the tissue. It's a really good idea to do.
No, I think that would be interesting, because obviously the primary tumor environment might be completely different to the metastatic one.
Yes.
And yes, anyway, sorry, in the meantime, there's another question here. So this is Josh Hadsley. Great talk, really interesting work shown here. Do you see changes in the MENA-specific gene regulation in tumors with increased stiffness or desmoplasia-heavy microenvironments?
Yeah, great question. So we've started to explore that a little. So we found that if we plate our cells on different substrates, if we make them engage integrins more, or if we put them on stiffer substrates, there is an upregulation of MENA and its recruitment into adhesion complexes as well. So we think that there is a mechanosensitive role beyond just regulating link complex function. So MENA appears to be more expressed. It appears to go to adhesion complexes more in those stiffer settings and when integrins are engaged, as compared to settings where we disengage integrins. So we think there is quite a keen link there between force in the microenvironment, the stiffness of the microenvironment, and how that mechanical force is transmitted into the nucleus. And MENA appears to be playing a multi-layered role in that response, But these are pilot experiments that we need to expand on further. But it appears to be going in that direction. Yes.
Thank you. Now, a question from Graham Murray. Many thanks for an excellent talk. Do you have any information regarding matrix metalloproteinases, especially the membrane-bound MMPs?
Great question. Did we saw some changes in secreted MMPs, but I don't think we saw any membrane-bound proteinases in our data sets. I will have to double check on that. But certainly we see some secreted enzymes that are involved in remodeling the matrix, and they change. So we think it's not just immune genes. There are other proteases and enzymes that are involved in remodeling the microenvironment that are being driven by both FAC and MENA. Actually, the expression of those genes are being driven by both those adhesion components. So I think it is a broader kind of remodeling of the matrix and the microenvironment involved, not just an immune microenvironment, but also a structural remodeling of the microenvironment. So we're following that up now and also developing a lung model to do that as well. So we'll be interested to see how that pans out. But yes, I think that is the case.
Thank you. So I can't see any further questions. Could I ask what would be your sort of immediate next steps or next projects that you're considering or are already in progress?
So I think going back to the mechanosensing element, I think that's a really important mechanism that we really need to tease apart in much greater detail. So clearly in a pathologically stiff tissue, adhesion receptors are going to be sensing those changes in biophysical properties of the tissue. So we think it's going to be a really important potential signaling pathway to understand that mechanism of likely integrins and other receptors sensing that change, transmitting that change in biophysical properties through the cytoskeleton to the nucleus. How is MENA doing that? How is it being driven to relocalize to the nuclear envelope? We know that it's in cases of cellular stress, this appears to be happening, and the same for FAC indeed. So things like oncogenic stress, mitogenic stress, oxidative stress appear to be triggering this relocalization of adhesion proteins towards or into the nucleus. So the mechanism for that is completely unknown. We don't know how those proteins are being trafficked there, if they're being taken, is it being driven through FAC's NLS via importins, and then there are passengers that go with it. We don't understand that mechanism at all. Is there vesicular trafficking involved? So that's something we're really keen to dissect. Are there indeed two separate pools of adhesion proteins? Is there an adhesion relevant pool and a separate pool, or are those pools interacting and transmitting messages from the cell periphery to the nucleus? Right. So these are kind of questions we're really keen to understand in the context of a stiffening tissue. So we're developing approaches to try and mechanically interrogate the cells with compression, with squeezing, with forcing cells to migrate through small pores and seeing how can the nucleus adapt to those changes if MENA isn't there to reinforce that connection to the cytoskeleton, for example. And that has implications for invasion as well, when tumor cells have to squeeze through pores in the matrix to invade. So is MENA important for controlling that architecture and infrastructure of the nucleus more broadly, given that it is clearly connecting the cytoskeleton to the nucleus? And those physical changes in the nucleus are really important for how chromatin is regulated, the organization of chromatin within the nucleus too. So it's a combination of the signaling that's involved and how forces are being imparted onto the cell and onto the nucleus to control gene expression and also tumor cell invasion. Right.
Well, fascinating. There's so much still to do, isn't there? Really, yeah. It seems to be never ending. Yeah. Okay, well, I think there are no further questions here. I don't have any further ones noted. So with that, I'd like to thank you very, very much, Adam, for your fantastic lecture this morning. And also I'd like to thank everybody for those who are online, who have attended this session live, and those who will be reviewing it in playback via YouTube. So I'd just like to let everybody know that the Grand Round lectures will be recommencing in 2026 in February. And this is our first lecturer is Professor Jan van der Thuizen, who is will be speaking about lung cancer and how the utilization of small lung cancer biopsies are important for diagnosis and also understanding the biology of the disease. And Jan is the president of the British division of the BDIAP. So with that, I'd like to thank you all very much and wish you all a very Merry Christmas. Also highlight that in January there will be the PathSoc winter meeting, which will be run over 2 days at the Royal Society of Medicine in London. So thank you very much. Thank you, Adam. And we'll see you next year. Thank you. Bye then.